So we had the added benefit in this field site of having a very specific and well-documented historical set of data for what was in the area, which of course isn't going to always be the case if you wanted to deploy this kind of fresh. So then you're looking at things like, if you look at your PCR positive control, if it's contaminated with something, or your negative control, sorry, if it's contaminated with something, which when you're working with vertebrates is usually you look at the read count, which is the amount of DNA in it, and you typically get rid of any hit or detection that has a read count lower than that. You do have to be careful with that because sometimes, again, you can get rid of true positives that way. That's like the very, very conservative way of doing it. There's other ways that are a little bit more forgiving. But you can also look at range maps and stuff. So in one of our other experiments that we did in that field lab, we were getting detections of bats from Africa and Europe and stuff, and we're like, that's what? Turns out people had worked with those species before coming, and we think what was happening is we were getting secondary transfer on their gear. So it is very sensitive. So yeah, those bioinformatic checks are really important, and then replicates. So the white-winged vampire bat, we're pretty sure it's a true positive because it had very high read counts, and we sampled that roost 3 different times, and it was the main hit every single time. Does that answer your question? Okay.
So I guess I think eDNA is cool, but my I guess question that anytime I hear an eDNA talk is it's basically presence absence, right? Which is important.
Yeah.
But if you want to know how many white-winged vampires are there, that's a challenge. And I know there are ways to get around it. So I'm with regular eDNA, not everyone. So I'm curious, if you have any thoughts on that.
Yeah, that's. Oh yeah, the question is, so eDNA is often presence-absent data. Can you get the number of individuals, correct? Yeah. That's a really big debated question in the eDNA community, and everybody really wants the amount of DNA to mean something. And there is evidence that it can, but so in that the other study, we did look at that. So for us, there was a positive correlation between the number of bats in the room and the number of detections we got, but there were 3 outliers, ones with really high read counts compared to what they should have been with their abundance, and then one with really low. Those were species, the ones with really high were species that were very hard to tell apart. So my supervisor was basically taking them out right next to our samplers and looking really closely at their teeth and handling them a lot to figure out which was which. So they probably shed more DNA. Whereas the one that was really low was the common vampire bat, and you can just poke the bag and it hisses at you. You don't even need to take it out of the bag to know that it's a vampire bat. So there is some evidence of it, but I think at least for airborne eDNA, it's still too early to tell if we'd actually be able to do that. So the question was, do we have a sense of how long the DNA signature persists? There was a study done out of Texas that used the dust collectors, and it looked at 2-week intervals. So we know that it in most cases, isn't longer than 2 weeks. And again, that classroom field lab experiment that I was talking about earlier, we noticed that it was a couple of days. So right now, again, that's something that we want to find out, do a little bit more work specifically focusing on that. But for now, it's anywhere between 2 weeks and 3 days. We think it settles relatively quickly, though. But yeah. Okay, thank you.
Thank you, Nina. Next up, we have a presentation from Derek Morningstar and Toby Thorne, right? Yeah, on to tell us about the North American Bat Monitoring Program.
Okay, thank you. And my clicker. So to answer everyone's question, by the way, it's not a good bat conference until someone dresses ridiculously, okay? Everybody wondered about the shirt. Okay. So, so when we were kind of planning this conference, we wanted to have some representation or a talk about NABat. So I'm going to tell you what it is soon if you don't already know. And we invited the coordinators of it from the USGS to give a talk for us. And they said, we'd love to. And then they said, but we have another conference, sorry. So I kind of got nominated because I post data to it to tell you about it and to do something else here. So some of this information is mine and making things up, but I also stole a lot of their slides. So you're going to see some stuff that's information about it. So I want everyone to get their phone out right now. Everybody get your phone out. Okay, this is an interactive thing. While I'm going through this presentation, I want all of you to fill out that survey. Okay, so we're going to talk about what NABat data is. And we all want to know from you if you have any, if you want to post it, if you have hurdles, what can we do to help? Because we want to try to get more data into it. So that QR code is going to stay up there the whole presentation. So if you were slower pulling your phone out of your pocket, get it out now, and you can still do it. Okay, so the NABat monitoring program is an international tool that's mostly for North America, predominantly in the US, but there's quite a bit happening in Canada now, to try and survey bats across the continent and look at a high scale what's going on. And this is kind of an example of what you can sort of get out of it. They do these modeling things. This is based on acoustics everywhere, particularly for northern myotis in this case, saying, okay, what's the probabilistic, where are you going to find them now? And this is, I think it was 2019 this data was from. If it was 10 years or 15 years prior, you'd probably find them everywhere. But what you can see here is, so this blue line is like the distribution of northern myotis, and you can see there's some huge white gaps. So granted, we might have those lines wrong on the distribution, that we certainly aren't confident on where it actually goes to, but there's a huge gap of where we just don't have any data for a lot of Canada because people aren't really posting it. But that data exists. People are collecting data, so we can do it. So the NABat monitoring program is, as I was mentioning, so this is kind of their mission and their goals, is to come up with standardized protocols on how to collect acoustic data, but also capture data and colony count data, and use a general sampling design to do that, and then integrate all that data together to come up with big high-level models like I showed you. So they have lots of different methods, papers. People have been using that data. Some people that don't even collect the data of their own, it's public access data, you can go and get it. So this is kind of what they're trying to do to look at changes over time of bat populations. And so this is kind of another example of how many people have been contributing to it. So in 2019, there was a little over 100 projects on there. Get to 2022, we're like 700 projects, and even since then, there's been quite a few more added. So there's a lot of projects on here, right? And like I said, it's public access data, so anyone can go on there and request some data and do some interesting stuff. So you can see the data is definitely growing. So this is kind of what they have as a grid design. So probably the first people they thought they were reaching out to was like state managers and provincial-wide like government managers that were setting up a program to sample for bats in a grid pattern across, at first the US, now in Canada too. And so they devised this grid sampling design. Well, they didn't come up with it, but they're using it. It came from somewhere else. So they're trying to cover all the areas. And in a lot of places you have, particularly in the US, there's state biologists that are collecting this information. In Ontario, there's a couple people that have been doing a bit of it, but not as much. But then they said, well, why would we stop there? So they opened it up to anyone doing any project on bats anywhere. There's a way for you to upload that data that can contribute to it. So it's kind of like if you think of eBird or iNaturalist, anybody can go and add stuff to it. There is a process. You have to register and do it, but you can. So, of course, now we got to look at Ontario. So in Ontario, as you can see, there's not a lot going on. So each of these black squares is one of the grid cells where there's been some data contributed. And it's 24 projects from 5 organizations. 4 of them are my clients. So most of this I've put on. I'd like to see more people putting data on there. Toby has got a whole bunch of data that he's putting on too now. So we need to grow this as a community. So I'm sure should have 1,000 points here if everybody put their data in. That's what this survey is all about, to find out. So I'm going to kind of whip through some of these method stuff, but you can add stationary point data. So for those that don't know, that's when you set a bat detector out there and you leave it for a long time. Mobile transect, that's active monitoring data. That's when you have a detector on the roof of your car or you walk around with it. But there is also the option of adding colony count data and maternity count data. I think most people do acoustics, but you can also put capture and count data. So we're focused on the stationary. So some advice about getting some information on there. So if you have acoustic data in particular, you really should plan ahead. So you can add it after. I'm going to say that first, and then I'm going to say you should think about it first. Because there's a lot of little tricks that if you capture your data the right way, it makes it a lot easier later. So setting up a bat detector, I don't really want to get too much into the details, but you want to try to avoid cluttered spaces. Never stick it in the middle of the forest, right? Stick it on edges and open areas. That's what this next slide about. There was a question earlier about the detection range of a microphone. Very best case scenario, you might get 50 meters around the microphone. That's for a perfectly omnidirectional microphone, which most aren't. But there's some information that they provide in their protocols about good ways to set up bat detectors, like on the edge of a forest and parallel to it, but a few meters away, how you would set it up on a roost or near a stream or a pond and stuff like that. So if you're interested in methods on how to set up acoustics, you can look at the protocols that NABat has. And so there is a criteria for the data to set to be able to contribute to it. So they want you to take all kinds of information about what kind of detector you used, what your settings were, and it's a minimum of 4 consecutive nights in order to post passive data. So if you've only collected for a night or two, you won't be able to post it, and if you did different nights, it won't work. So you could have the whole season every other night, and I had a client who did that. I'm like, well, sorry, we can't take it. Anyway, so 4 consecutive nights is the minimum. But then, I have some that are 150 nights long. There's no maximum. You can keep posting. So it's really important to take good data where you set up your equipment, and I'm not going to go through this, but you should have a really good data sheet that works as a checklist, and make sure you do everything the same way every time, and you can document the information, because they're going to be asking for that metadata, which is how you set it up, where you set it up, the equipment you used, all these things. It also helps to troubleshoot if you run into trouble. And I put this a couple times. When you're dealing with metadata and putting it into here, the comma is your enemy, because they use comma delimited spreadsheets. So we enter it into a regular Excel spreadsheet and it all seems fine, and you put commas in your description of the habitat, and then it splits it out to 1,000 different columns, and you have to manually put, don't use a comma, just don't. So there's information that they want, as I was saying, minimum fields, there's required information. So remember, when somebody else goes to look at this and they're looking at 1,000 stations, they want to know a little bit about those different stations, right? If you were thinking and looking at it, how do you standardize what you're taking? So they want to know when and where you took it, what equipment you used, and there's some optional fields of, what was the orientation. To me, I wouldn't want these optional, I would want them to be required, but they say they're optional, about more of the context of where you put it. But I would say the more information you can get, the better. It helps when someone else looks at that data. So when you first come back with all your acoustic data, you've got to attribute your data. There's tools to do this, depending which software you're using. This is with the NABat platform. There's other ways to attribute data. The comma should never, ever be put in one of these, because it really messes things up. But the other thing I was going to point out is that it can be kind of frustrating. A lot of these things, like habitat type, I think there's 5 options. So we put these in lots of different habitats, and you have to pick one of those 5 options. So, be prepared that, if you have a paragraph long describing your habitat, you get 2 words here. So, there's some strict criteria that you're going to have to follow eventually. And just to remind you, the comma is your enemy. And then you can analyze your acoustics. Okay, I'm going to have to whip through here. I'm not going to get it all into how to analyze. That's a tricolored bat. It's a nice little bat. You export your data, and you have to export it in their format, but they have forms, so you can do that. Again, there's different software to do that. And then you upload your data to the website. And when you upload the data, so you first upload the bulk data. So that's CSV files that talk about the files you have, but not the actual WAV files. And then you upload your WAV files. That can be frustrating. So your metadata file, it all gets error checked automatically, so you spend a lot of time going back and taking out those commas and fixing all your columns and stuff like that. But then when you upload the WAV files, it can take a very long time if you have a lot of data. So the wordy slide here, there's a few different ways to do this. If you have small projects, and I just kind of threw 100 gigabytes as a bit of an example, or 100 nights or less or something like that, it's pretty easy to upload all that stuff. When you get really big data sets, like some of mine, I have several hundred nights of data and thousands upon thousands of files. That takes weeks to upload. But you can either upload it all, and then that data, by the way, your WAV files are backed up in another way. So if you have a way to back it up that way, you can just put the metadata file that talks about your files and then kind of promise to keep your data safe somewhere, and when somebody wants to look at it, they do the request through you. So then you don't have to post it, but you just have to keep a copy of that data. And there's another option, which is called the BAT AMP format. It's actually from a different site, the Acoustic Monitoring Protocol. I forget the right word, but anyways, that basically is just a spreadsheet that says a summary of the passes per night by species. So it's not even talking about individual files, it's an even higher level one that you can put so that your data at least shows up in those, data summaries, and then somebody knows that data was taken there and they can ask you for more details on that. So I've already said kind of a lot of this advice. Plan ahead, take good field notes, stay organized with all that data. The comma is your enemy. And don't forget to vet all your acoustic data. You should probably vet it before it goes onto this. It can take a long for formatting, but it's possible to do it. And it can take even longer to load those files, but once you get it all in there, there's the ability to have some really strong, powerful, high-level tools about bat populations. So I think that's the end of my time, so thank you. Do we have time for questions? I guess we have time for a question or two.
So for data that people collect, like batwatch.ca, is that worthwhile looking into contributing to NABat? Or
So yeah, so there's overlapping different locations. So batwatch.ca, I believe, is more about roost counts and colony counts, which this has too. I don't know why they haven't gotten together, but I don't know if they share the data. So honestly, I don't know the answer, but it is similar data. This is a North American scale. Batwatch is more of a Canadian scale. I don't know the right answer to that, but they should probably get in the same room and figure it out. Any other questions? Yeah.
speaking of people getting in the same room, is there much of any communication with NABat and, say, the provincial government?
Yeah, so I think. So, okay, sorry, yeah. So the question was whether our province is doing anything for NABat from the ministry. And so the short answer is yes, but I think it's limited. So I know Mark Browning was doing some driving transects. I think they have some acoustic data. I haven't actually met you, but I was trying to see if there's any other government people here. So I think they've done a little bit at the provincial level. At the federal level, they've been doing data collection. I analyze the data for them, so most of that data is actually from the federal government, or a lot of that is. So there is some coming from Canadian government and provincial government, but it's nowhere near what they're doing in the States. So really, there's all kinds of other people collecting data that would be a lot more than that. So, yeah. Any other questions or one more?
Is there an equivalent that's a global monitoring program?
That's a good question, and to my knowledge, no, not at this level, not that kind of data. I know there's a organization in Southeast Asia that does something like this, or they're trying to. There's other organizations, and so like there's NABat for us, or NABat, but NASBR is a community. There's other bat communities in other parts of the world that I know of that probably do a bit of this, but I'm not sure if they have this level of organization of data repository. Like, I mean, it's probably hundreds, just imagining all that data, hundreds and hundreds of terabytes of data that somehow they're storing. But yeah, it's huge. Do you know, Liam?
Well, it's not at this grain, but GBATnet has a project right now to
Okay.
gather global data sets.
Yeah, right, okay. Cool, yeah.
See how it comes together, but they're working.
Right, good point. Yeah, so GBATnet, look that up. That's the global version. Yeah, and I think it's not as detailed, not quite as refined, but
Network all the different.
yeah.
parts, and NABat is one of them.
Yeah, so you can find Google GBATnet and you can know more about the global level. I think that's all the time I have, right? I'm horrible at repeating the question. It was whether there's a global option for putting data in. Oh, we have more time. Is there any, yeah?
So do you need to individually ID every wave file that you want to upload? Or is there a way to say, I found these 3 different bat species, here's all the data, there's these in.
Did you fill out the survey?
I'm in the midst of it.
So that question is asked in there. So the short answer is, ideally, yes, they would like data that's already been analyzed, and vetted, but it's a public or top repository, so they'll take all forms of data, and they're working on ways to do analysis on there. They're working on AI programs that's supposed to be better than the auto analysis. I don't think it's really getting there, but they have a whole team of people that are trying to find ways to deal with data that no one had a chance to analyze. They're also trying to find a way to offer, maybe some people have time to do analysis for you. You can put it on, other people can do analysis. Like, they're trying to coordinate all those things at NABat. So I didn't do a good job of talking about it, but that's another thing that they're trying to do. So it's not just finished data. You have the opportunity to at least let them know about other data.
As a quick follow-up, if I had vetted, let's say 20%, would they rather I just upload the 20% I vetted or 100% with the 80 that I haven't
So that question was, it started with, can you upload data that hasn't been ID'd or vetted? And then the follow-up was, what if it's partly vetted or ID'd? And honestly, I don't have the right answer for that, but because I'm not the one who deals with it. But I think you can always communicate, so that the team that couldn't come today, the NABat coordinators, are really helpful. So you can contact them, go to the website, and they will answer all those questions for you, and they really want to get your data, so they will find a way to help you. So, yeah. I think that's it. Okay.
Great. Thanks, Derek. Okay.